introduce our next distinguished speaker, Dr. Kjell Stenberg, Venture Partner of Nodes Advisors. Dr. Kjell is a partner at Nodes Advisor and a member of the Corporate Finance Group. Dr. Kjell is involved in the firm's corporate finance and capital raising activities with a focus on the European market. Prior to joining Nodes Advisor, Dr. Kjell co-founded and served on the boards of successful biotech companies such as Galecto Biotech, Aptahem, and Saison, the latter of which he became the CEO. These three companies combined raised more than $100 million USD in the first 5 years. Dr. Kjell's large pharma background spans bioinvesting with Medwell Capital and managing projects in cancer, infectious disease, neuroinflammatory disorders, and antiviral therapy at AstraZeneca. So without any further ado, I want to give the stage to Dr. Kjell. Please.
Thank you very much for that great introduction. I hope that everyone has had a good lunch and is ready to hear some thoughts on accelerating drug discovery. I have a great, turn to the first slide. You have made it, the presentation is just wonderful. I just want to summarize that I've been in large pharma and biotech for quite some time. And I left large pharma mainly due to the difficulty to be innovative and really make good drug discovery in a large organization. And I turned 2001, I turned to biopharma companies in Sweden, in Denmark, and in North America. Nodes Advisors, I represent Nodes at this meeting because the Nodes is heavily engaged in drug discovery and financing. It has just launched a program in an AI. So you can see that at that Nodes page if you're interested. I've divided my speech in 3 parts. First, a short overview of drug discovery, what has been done and what and why has failed. I also want to speak on one way to discover a new drug that is shortcutting the long and tedious way of going from an academic research to a development stage. The drug discovery is really what is really hampered drug discovery and drug production. The production of new drugs has been heavily reduced by the inability of large pharma and also biotech to really find the new compounds that we need to find better medicines. And the reason, I think we can blame nature for that, because it's often very, very complicated biological backgrounds of the diseases. They can be caused by a series of different deficiencies and malfunctions. And drugs typically also impact several different types of biological targets. So if you're developing a drug, you don't really know what's going to happen. You develop it for one biological target, and then you find out in safety studies at least that it doesn't really work the way you thought. It's maybe toxic. You have to take that into account. Or it's also what has been happening is also the inadequacy in really making what we called in the 1990s for docking experiments. That is to see the panel on the right side of the slide just denotes the way that what we in drug discovery in the 90s had in front of us daytime. We were exploring different moieties in a new drug, and we're trying to optimize the structure of the drug to really to increase efficacy, increase selectivity, and also decrease toxicity. And the next layer also increase, let's say, bioavailability. The process of drug discovery can be denoted in this time. If you look back 100 years for penicillin, this was purely a macroscopic scientific observation by Fleming in the 1927s. When I started in drug discovery, I was working in antiviral therapy, and we were discovering new drugs for retrovirus therapy. And that was a collaboration by chemists, medical chemists and virologists. It was a tedious job, sort of the same thing I showed on the last slide. Then came the era of high-throughput screening combinatorial chemistry. And this was very one built big, large factories. I met a Pfizer representative at that time in the late 90s, and they were showing big pictures of huge industries. What is coming now and what we are working towards is using AI, precision SRA, and repositioning of existing drug to accelerate development of new drugs for man. If we take that one step by one, the pre-SAR, the serendipity area, in the, let's say, up to the 1960s, most drugs were found using skilled chemists and skilled biologists. Penicillin, we talked about, insulin was an analog. It was really the same compound as the biological effector. Losec was discovered in the 70s by Astra, the company I worked for. And this was half serendipity, half scientific experience, taking into account a valid biological target. There are a lot of macroscopic exercises that led up to this. The next happened, the combinatorial chemistry in HES. These big factories, although they processed thousands and thousands and thousands of compound, they didn't really come up with very much that was superior to what was really in the pipeline so far. And it was heavily, very, very expensive, very, very difficult to run logistically. So factory approach was phased out in the beginning of the 2000s. I founded a company in 2001 in Denmark in combinatorial chemistry, and I see over the 3 years I worked in Denmark with this combinatorial company, it was a fading interest from industry. It's very, very sad for the investors, but still it was the reality. What's happening now is the possibility to use AI and develop new AIs, algorithms and ways to solve data handling, to be able to handle a large amount of drug data. And I think that if we are doing this very neatly, we can really maximum the use of accumulated knowledge, accumulated data to really go forward to find new and better drugs. Repositioning is, I'm coming back into that as well, it's using failed compounds. The way that it works in industries, if you're having a compound developed over years and years and years, maybe decades, in the academic sense, it will go into phase 1 and it go into phase 2. And if it fails in the clinical phase 2 study, it's dropped, it's left, it's abandoned, and no one really wants to see that drug again. Me and a colleague in the US, we have been taking advantage of that together with a number of other companies to see whether we're able to take this drug that have a failed drug for a new disease where it's more functional. So this is the three parts: repositioning and how we use it and the process to market of the product. It's when the drug that everyone thinks that it works well in the in vitro, it works well in animal models. You have disease models, and it works well. A clinical study is warranted, and the compound is really passing phase 1. It's not toxic. It's safe for man. It will go into phase 2. You have spent a huge amount of money at that stage. About half of these compounds fail that go into clinical trials. 54% of the number of compounds that go into phase 2 are survive phase 2, and the other one, 46%, are just discarded. This is fairly reasonably close in time. It's only from 2004 people got to recognize that we have to do drug discovery in a better way, because the attrition rates, the failure rates in pharma were escalating and costs were escalating. Repositioning of drug is a low failure rate because you don't have failures for safety reasons usually. Because you are using the same compound, usually the same dosing regime, and you give it to another cohort of patients. But you can of course fail in activity, but the statistical probability of success is higher. Repositioned drug currently contribute to 25% of the annual revenue rates in pharma companies. So 1/4 of the money that goes into the system comes from these drugs. So it's not a minor thing. And if you take the top 10 blockbusters, they have been selling 12.9 billion US up till 2015. So it's a really profitable area. And deals are made. For example, Fetritinib by Targegene, they sold that drug to Calgene for 4.3 billion last year. So it's really a profitable business. Why can we repositioning drugs? What have they missed? People who originally developed that drug. That's because that biological targets are often shared in different diseases, meaning that you don't really know sometimes where you're going to drug, and the drug is also pleiotropic. For example, on the first side, the Parkinson's disease and Alzheimer's disease share 48 genes and 4 signaling pathways, and same drug may work or might therefore work in different diseases. So if you have a drug with addressing 1 or 2 or 3 common genes in Alzheimer and Parkinson, you are likely to find some activity in both diseases. It's also a pleiotropic response to the drugs is that, for example, as acetylsalicylic acid or aspirin in the US, you can inhibit an enzyme called COX-2 and to get an analgesic effect. But if you just lower the concentration, it will inhibit a enzyme COX-1, and that is for platelet inhibition. So the same drug can be given to relieve pain at higher doses, and it also can prevent heart failure by preventing coagulation in patients who are prone to do that. So the big secret of how to repositioning drugs is to understand in detail disease physiology and to identify these drugs, the pleiotropic drugs, that can also act on the target in the new disease in a good way. Personally, I think that this is something that will be producing more drugs as we go, and using AI to really support that process is really going to make potential to make a leap in the area. Repositioning, again, why is it quicker? As I told you that it was safer. That's safe. It's not as risky for developing a drug, for a repositioning drug, but it can also avoid the long process. The chevrons on the slide goes from preclinical to NDA, and if you add up these years, it's half of some people's lifetime. So this is a very costly, very slow process, and that's why it's produced so little if you follow that very strictly. What we do in repositioning is to take the drug, identify the disease, and run a phase 2 trial, a proof of concept trial. And that will take, depending on the disease, it can take from 1.5 years to 4 years, but around 2 years in a mean. So after 2 years, you can really demonstrate in man that you have a drug that is likely to work and become a product. And in that case, you can really find a partner for doing the phase 3 and also do the marketing efforts at the end of the trial. So it's a very cost-effective and easy way once you find the right compound or the right disease. And I have a company called Actina I'm currently financing together with my partner to really do some business in this area. In general, I think that we can have the factors that will really have a potential to accelerate drug discovery is the structure activity data. I think that the more higher resolution in the technologies we have, and the more ability to characterize the properties of this drug, also the solubility, stability type of properties, which is important for a successful drug. And use of the right or the relevant disease models from start to end to really increase the predictivity to man for the drug in question. And also a good data collection. A systematic data collection means that you can use data from different systems and compare them. And I'm just hinting on the cleaning procedure. I have my thesis from the Karolinska Institute, and you usually don't publish things that are negative. But I think that some in the clinical trial setting, people publish extensively on negative data sometimes. And I think that's really a good thing. And I would like to see that also in let's say, basic science. Improving AI tools. I'm not an expert in AI, but I see that as a very, very powerful technology to be able to analyze objectively a huge amount of different types of data from different sources. And a man's or a woman's brain is not just able to really comprehend the enormous amount of data there are nowadays. In the 1970s or 80s, the amount of data was manageable for many companies and even for many individuals to keep track of, but it's completely impossible today. So I'm looking forward to powerful and strong AI tools to do a lot of things that people did in their brains and their minds so a few decades ago, and really come up with new connections, new combinations of data that generate new drugs. But still, we can't really rely. We will never find. I hope we will, but I don't think it's feasible to have an automatic system to really discover new drugs. I don't think that's really doable. So what I see is, if I would love scientists, both basic science, animal science, and in vitro science scientists, and clinical scientists as well, together with pharmacists and medical people, working together with these AI systems to really make use of optimal use of the data system. So what we can do is we can find efficacy, safety, distribution, excretion, stability. We should have all these types of data in the same database and same accessible for the AI. I think that we can find really novel chemical structures that are markedly better than those of today. We can in that sense, with better drugs, we can also increase success rate in clinical trials, and we accelerate the process in the scientific, in the clinical sense as well, in the clinical sense. And we will shorten the time to approve product. And that's very important for pharma industry, and it's also important for investors in the biopharma world to really come up with investments that can be, let's say, give return on investments within reasonable time. We also have once again remind that we can sometimes have a very short development program using drugs that are well known for other diseases, and that is really a very obvious shortcut. It only requires people who have a good sense of disease pathology and drug insights. And this is where I want to end, and thank you for your attention, and I'm open for questions.
Thank you so much, Dr. Kjell, for your amazing presentation. It was very insightful, especially the chart you showed in terms of the progress in years. That can be very useful. So I will open up the floor now to any questions, comments, or remarks from the audience. So we'll take a moment here if anyone has anything. Again, Nodes Advisors is a corporate finance firm that works closely with life science and healthcare companies, mainly focusing on breakthrough and novel technologies for life sciences. So if you're an entrepreneur or interested in learning more, we can definitely connect you with Dr. Kjell.
Well, can I just make a mention, one concluding mention? If people are working on drug discovery and you're not feeling very motivated, you think it's difficult, think about that the large pharma and the biotechs are dependent on intelligent, brilliant people doing a great job in discovery to really pave the way for clinical trials and new drug for us, the mankind.
Absolutely. No, that's a really good point. And yeah, Dr. Shell will be back in a few hours at the panel discussion as well, giving his insight
Yeah.
there too. So again, thank you so much for your presentation.
Thank you for allowing me to speak.
You're welcome. Bye.